amylogenesis, bone homeostasis, and other biochemical functions are influenced by genetic as well as epigenetic factors. However, the underlying molecular mechanism needs to be explored. Despite factors such as environmental factors, dietary intake of fluoride, and other minerals being constant, the documentary evidence shows that genetic and epigenetic plays a key role in inducing these toxic effects of fluorosis. The Kolar district and its neighboring areas have been weighed down by the crippling of maladies of fluorosis, diabetes, and cancer. In this particular district, initially, many of the clinicians are wondering why at the early age group, even in second decade or third decade, people are becoming crippled with joint pains and presenting with unhealthy bony disorders and dental disorders. Now it has been more evident it is because of the fluorosis. The Department of Biochemistry, with its thirst area of fluorosis, is poised to provide an insight on fluoride and its linked genetic disease, with special emphasis on diabetic mellitus and malignancy related disorders. With these few remarks, I wish the biochemistry department and the organizers of this webinar a grand success. Hope all of you will be enriched with the more inputs about the fluorosis related disorders by the end of the webinar. I thank the organizers for giving me this opportunity to share my remarks with you all. Thank you.
Thank you, sir.
Thank you, sir. Now I request Dr. K. N. Shashidhar, sir, to give overview of fluoride and its link with genetic diseases, with a special emphasis on diabetes mellitus and cancer. In-house studies report. Over to you, sir.
A very good afternoon to one and all. I thank Dr. Soumya for giving an introduction about myself, the department, the Fluorosis Research Referral Lab, and I am extremely thankful to our Dean, Dr. P. N. Sriramulu sir, the principal and the Dean, Dr. P. N. Sriramlu sir, for recognizing the work that the Department of Biochemistry is doing and encouraging us to go ahead and get the Center of Excellence to the Department of Biochemistry, the Fluorosis Research and Referral Lab. So I'm thankful to you, sir. So with this, let me welcome you all to this webinar. And the title of the webinar is Overview of Fluoride and its link with genetic diseases, with a special emphasis on the diabetes mellitus and cancer. I will be speaking on the in-house study report. So we have done exclusive studies on the fluorosis and its mitigation. And before I start, I would like to acknowledge and thank my colleague, Dr. Harish, Dr. Soumya, Dr. Sai Deepika, Dr. Munilakshmi, and Mr. Bhuvaneshwar for helping in taking the fluorosis project ahead. The main objectives of today's webinar is to provide an overview of the fluorosis, to highlight the mechanism evincing the different types of fluorosis, describe the variables bridging the epigenetic and genetic factors in fluorosis and its linked disorders. As we all know, the fluoride is the 13th most abundant element in the Earth's crust. It exists as fluorine in nature and it is anion in character. Fluoride exists as a fluorite ion in the nature. Fluoride is one of the few elements that have been shown to cause significant effects. The major sources of the fluoride includes the drinking water, the air, where it exists as a fluorine gas, and the dental products, where it exists as a fluorite ion, the food and beverages, where it exists as fluoride. So this has been studied extensively by my colleague, Dr. Dina Mendez, and we have written a review on this. Myself and Dr. Dina Mendez were involved in this study, and we had done chronic fluorosis, a disease of concern, and it has got published also. The fluoride, initially it was thought that it is involved in the skeletal and dental fluorosis. Now it has been of late, it has been observed that it is an inducer of the gene toxicity, which accelerates the genetic disease as well as the cancer process. The global disease burden of the fluorosis is very high. This has been documented by our in-house study, which has been conducted by Dr. Deepika, myself, and our Dean, Sriramlu sir. We have conducted a study on the global disease burden of the fluorosis, in which the Kola district has been observed. Kola district lies in the eastern part of the Karnataka, where it has been observed that the water table has gone beyond 1,300 feet, where the fluoride content is very high. It has been documented that the ppm of the fluoride in the water has crossed beyond 800. So this created interest in us, and this study has been published in Journal of Evolution Medicine Dental Science, which is a Scopus Index journal. The possible mechanisms that we could observe was the fluoride reacts with the genes by altering the RNA and DNA with chromosomal aberrations and cytotoxicity, which in turn will end up with the diseases and aplasia. Initially, when the fluoride was thought, it was thought that it is a molecule of interest and it was very much essential. Yes, no doubt, this is a molecule of very much interest. It acts as a double-edged sword. Initially, it was observed that due to the deficiency, it has been added to the toothpaste and various other food as an additives. And it has been observed that as the water table has gone deeper and deeper, it had an impact on the health. And it has been observed that the fluoride, in addition to the dental, it also had skeletal. And of late, it has been observed that even the non-skeletal organs are also involved with the fluoride and its impact. The mechanisms of skeletal fluorosis includes the metabolic bone disease, with osteosclerosis as the major clinical manifestation, followed by osteoporosis, osteopenia, and osteomalacia. The fluoride substitutes for the hydroxyl group in hydroxyapatite, forming fluoroapatite. And the bone metabolism includes the process of osteoblast, forming bone, and osteoclast, degrading bone. Fluoride has an effect on bone mineral, bone cells, and bone architecture. We have observed the mechanisms of dental fluorosis and we have published it in a study titled Mysterious Mineral, the Fluoride. This study has been conducted by Dr. Deepika, myself and P.N. Sriramlu, and this has been published in Journal of Evolution Medical Dental Science. The action of fluoride on proteinases across the dental fluorosis is inhibitory, resulting in precipitation of amylogenin on the surface of dentin, causing plaque formation. And it is the proteinases that are very much essential, essential for enamel organ matrix synthesis is going to be defective with the effect of fluoride. The accumulated amylogenin may be due to improper protein folding by endoplasmic reticulum, which is under stress due to fluorosis. The protein folding failure is one of the major reasons today for ample number of disorders resulting due to fluorosis. To say a few things about the non-skeletal fluorosis, which is the disease of concern of late, and this is the newer thing that has been observed across the globe, and this has created us in taking up the fluorosis research referral lab, and we have started with a small laboratory, and which has grown up to the center of excellence as on date. The fluoride was observed to damage DNA of the cells in organs, which are involved in the metabolism, resulting in mutation of the signaling pathway of the enzymes and proteins, leading to metabolic damage and depreciation in normal cell functioning. Again, this study has also been quoted as before. It has been observed that in addition to these things, even the fetus are also getting affected with the high exposure of fluoride across the maternal age group. The fluoride has been found in the amniotic fluid in the pregnant women living in areas of high fluoride concentration, and this result in fetuses being exposed to elevated fluoride levels and premature deliveries, miscarriages, and so many other abnormalities, and even insulin resistance has been observed with respect to the high exposure to the fluoride. The fluoride defects is observed as it can cross the placental barrier into the fetal tissue. It affects the fetal brain tissue and cause neurological damage as well as neuronal degeneration. It also causes reduced secretion of the norepinephrine. Next, the GIT implications of the fluoride. High concentrations of the fluoride react with the HCl in the stomach to form hydrogen fluoride. Excessive formation of hydrofluoric acid results in irritation of the gastric mucosa, leading to ulcer, finally to the cancer. Fluoride stimulates secretion of the HCl, diminishes the blood supply in the stomach lining, causes death of the epithelial cells in the GIT, causing baldness of the GIT. The neurotoxic effects that has been observed includes the excessive fluoride crosses the blood-brain barrier and accumulate in the brain, causing dysfunction of the central nervous system. CNS during development is highly sensitive to the influence of the fluorine due to its weakened protective mechanisms. The studies have showed that children in high fluoride areas had significantly lower IQ. We have taken up a study to measure the intelligent quotient scores than those who lived in low fluoride areas. And we are comparing the two groups, and even we are planning to conduct a clinical trial based on the natural ingredients that are available in this local area, where it can be used extensively. Routinely, those things are consumed, and we would like to extract the ingredients which are repellent to the fluoride or inhibits the effect of the fluoride. Once that is done, the data will be published, and we are planning to give recommendations to the government of Karnataka as well as government of India. The reproductive system, the lower fertility rates which has been observed in this population are in general the high fluoride exposure population are documented with increased fluoride. Prolonged fluoride exposure is associated with increased FSH and LH and decreased estrogen levels. This is a contributory effect in addition to various other factors that are involved in the low fertility rates. It affects the spermatogenesis, oligospermia, sperm differentiation, and impairment of maturation, increase in chromosomal aberrations in primary testicular cells, and sperm deformity. Of late, it has been shown that exposure to fluoride can also alter the blood-testis barrier, where the circulation to the testis is decreased and leading to oxidative stress in the testicular cells. The cell apoptosis is one early sign of genotoxic damage that has been observed in the mature testis and plays critical role in spermatogenesis. Cardiovascular system. Of late, it has been observed in the local population that majority of the people at younger age are suffering from cardiovascular diseases, particularly the atherosclerotic vascular diseases. The ECG of the patients with the dental fluorosis has depicted abnormal heart rhythms. Fluoride can accumulate in the cardiovascular system, resulting in arterial calcifications, elastic properties of the ascending aorta disruption, and ventricular diastolic function. We are planning to take up a study in association with the radiology department and cardiology department to take it further. With respect to the endocrine system, insulin secretion and diabetes, it has been observed that isolated islets of Langerhans cells show that the basal as well as glucose-stimulated insulin is repressed as the fluoride concentration increases. The fluoride and advanced glycation end products lead to development of microvascular complication in patients with diabetes, leading to diabetic kidney disease. We have done 3 studies based on this one, and we have got a positive correlation with respect to the fluoride exposure and insulin resistance. Effects on thyroid gland, also we have done a study, and it has been observed to have an impact on the thyroid gland also. Fluoride, as I mentioned before, it is a more electronegative than the iodine, easily displaces the iodine and affects the thyroid gland. It is regulated by a negative feedback mechanism, and when pituitary gland senses drop in free T3 levels in circulations, it releases more TSH, stimulates the thyroid gland to accelerate production of T4. Fluoride interfere with the activity of enzymes of deiodination, iodothyronine deiodinases. Fluoride increases the concentration of TSH and decreases the concentration of T3 and T4 hormones, leading to hypothyroid conditions. With respect to the cancer, we have observed in-house, we have done a few studies, particularly with respect to the oral cancer. We are planning to take up extensively with respect to various other cancers such as the cervical cancer, ovarian cancer, carcinoma of breast, and various other diseases, CA stomach and other things. Fluoride, it has been observed to affect bone formation and mineralization, which are influenced by the genetics. The association between genetic polymorphisms in candidate genes and the susceptibility in the development of fluorosis has been documented. The examples of the candidate genes which are associated with human dental fluorosis and skeletal fluorosis are collagen type alpha 2, ESR, that is estrogen receptor, and vitamin D receptor binding proteins. We have taken up a study and we have completed a PhD thesis on the vitamin D receptor studies. The overview of the fluorosis, it has got impact on the genetic susceptibility and various other neurological, from head to toe, we could observe various defects with respect to the high fluoride exposure. Our in-house study, we have taken up fluoride with the linked disorders with respect to the reproductive health, CNS excitotoxicity, renal disorders, diabetes mellitus, cardiovascular disease risk, and thyroid dysfunction. These are some of the major areas which we are covering up with respect to the fluorosis research and referral lab, which is identified as the center of excellence by our academy. The fluorosis research and referral lab has been notified, and the major scientists who are involved with respect to the fluorosis research and mitigation as thrust area of Department of Biochemistry of our academy are Dr. Shashidhar, Dr. Dina Mendes, Dr. Muni Lakshmi, Dr. Harish, Dr. Soumya, Dr. Sai Deepika, and Mr. Bhuvneshwar Yadav. We have got the fluorosis research and referral laboratory, which is beneficial. It has been observed that fluoride is beneficial in small amounts, but toxic in large amounts. This is the overall view that we have observed as on date, and it is linked with various other disorders. So this is our lab with various other activities that are being carried out. We are doing the diagnostic work. We are educating and creating awareness among the population. We are doing research on the fluoride. We are also involved in the community health services in association with various other departments. Our in-house studies have published data, and we have many independent publications which have been accepted and yet to be published. We have done more than 6 PhD thesis work that has been completed and submitted. Many MD and MS dissertation reports, which the numbers I will be giving out, and the outcome of these studies also I will be highlighting. Preliminary analysis of the ongoing research has got a positive note with respect to the fluoride exposure. We have 7 in-house fluorosis projects. So the first one which I am going to mention here is the estimation of fluoride and certain one in patients with diabetic nephropathy in Kolar district of Karnataka, India. This is a PhD thesis work. This has been carried out by Dr. Sai Deepika with myself as a guide, with community medicine and medicine professor as co-guides. And we have observed that the fluoride has got increased exposure to fluoride has got an impact on the diabetic status of the people, ultimately leading to earlier occurrence of the diabetic nephropathy. Correlation of serum and urine fluoride with anemia and maternal perinatal outcome in pregnancy. This study has been done by Dr. Sneha Singh and with myself as a co-guide. This is a PG MS OBG dissertation, and we have observed that there is a strong correlation of serum and urine fluoride with anemia and maternal perinatal outcome in pregnancy. It has been observed that in addition to the iron deficiency anemia, even vitamin B12 and folic acid deficiency is also being observed with respect to high exposure to the fluoride. The molecular mechanism, how the vitamin B12 and folic acid are involved, needs to be established yet. And we are working out on that one. We have another in-house study. This is our academy-funded project. There are 2 projects which I wish to mention here. One is association of plasma fluoride levels with occurrence in pre-diabetes in 3 endemic villages of Kolar district. These areas, which are around 25 to 30 kilometers from the center of our institution, we have observed that these 3 areas, there is a high fluoride concentration in the drinking water, and it has been observed that the transportation of water from the source to the utility, the fluoride concentration is differing. This has created interest in us, like, does the plastic pipes that has been used or the PVC pipes that has been used, is it attracting the fluoride and reducing the fluoride at this from the source to the consumption is one of the interesting factors that we have observed with respect to this study. As I have mentioned, so the Moringa oleifera leaves, we are starting with a clinical trial based on this one. So the drumstick leaves on the hematological profile and histomorphology of brain, liver, spleen, and kidneys of fluorosis in the affected rats. And we have got a positive note with respect to this one. And we plan and we wish to extend this study with respect to the human beings as well as school children. This study has been done by the PhD scholar of Department of Physiology, guided by head of the department, Dr. Vinta Shankar of Physiology, myself, and pathology professor, Dr. Hemlata. We have one more study, knowledge and awareness about fluorosis in rural project, Masters of Science project, impact of increased fluoride intake on function of thyroid hormones in Kolar district, an area endemic for fluorosis. This study was guided by Dr. Dina Mendez, one of the co-associate of the Fluorosis Research and Referral Lab. And we have observed, and this study has been sent for publication, and it is embedded publication, and it has been accepted actually in Scopus Index Journal. And it has been observed that the increased fluoride intake has got an impact on the thyroid hormones, particularly the T3 and T4 levels are reduced with an elevated TSH levels. Next study that we have done is the association of vitamin D binding protein in diabetic nephropathy. This is a cross-sectional analytical study. This study has been done by Mr. Bhuneshwar Yadav. This is a PhD thesis work. The work has been accepted, and shortly he will be making a defense presentation for the award of the PhD degree. This study has been conducted under the guidance of Dr. Shashidhar K. and that's myself. In-house fluorosis projects, we have got 5 projects, and I will be presenting the midterm report here because till the report is accepted, I cannot I cannot present the final report. We have observed, we have done a study on fluoride, a missing link between diabetes type 2 and microvascular complications, a tertiary care rural hospital case control study. We have observed that in addition to the nephropathy, even the retinopathy, cataract has got an impact on high fluoride exposure, where we have observed that the high fluoride exposure has got an impact on the magnesium and manganese ions, and this is the one that is causing the inhibition of the glycolysis, and glycolysis glucose is taking an alternative pathway, entering into the high polyol pathway, that is the high concentration of sorbitol is observed with respect to the exposure to the high fluoride levels. Role of fluoride and oxidative stress parameters in senile cataract, a tertiary care hospital study has been conducted by is in the process. Dr. Sangeeta T, ophthalmology, PhD thesis work, and myself is the co-guide for this one. And we have, the study is in progress, and the intermediate report needs to be published yet. We have one more study that has been conducted on the histomorphological change and glutamate levels in fluoride and arsenic-induced injury to hippocampus and cerebral cortex in Wister rats, and ameliorating effects of ginkgo biloba. And the results are favoring that high fluoride exposure will lead to histomorphological change, as well as the glutamate levels in the fluoride and arsenic-induced injury in the hippocampus and cerebral cortex in the Wister rats. The serum biomarker, serum fluoride levels estimation with histopathological changes in the placenta in pre-eclamptic women. This is a PhD thesis work. This has been submitted for final review. Once this is done, the study will be ready for the open defense. And we have got a positive result with respect to this, and an article, 2 articles has been published with respect to this. In-house fluorosis project, which is ongoing, is the influence of the fluorosis on genetic predisposition due to single nucleotide polymorphism of LOX1 gene in pseudo-exfoliation syndrome. And we have got favorable results with respect to this. We could see the mutations with respect to this study. We have published 8 articles. One is the mysterious mineral fluoride. And this is a PhD review article, which has got published in 2019. Estimation of serum fluoride and renal parameters in diabetic nephropathy, a facility-based observational case control study. This is a student-related project. This is an STS ICMR 2020 article, which has got published. And we have observed that the serum fluoride has got an impact on the diabetes. Diabetes, in turn, will lead to early damage to the renal as well as the retinal cells. A cross-sectional study of serum and urine fluoride in diabetes in fluoride-exposed population. This is a PhD work, and this has got published. And we have done a study on the estimation of fluoride and SERTIN1 in patients with diabetic nephropathy in Kolar district of Karnataka. We have taken the study group within 15 kilometers of the radius from the hospital, and we have observed, and we wish to extend the study up to 60 kilometers in the next phase. Serum fluoride levels as a biomarker in pre-eclamptic women, a case-controlled tertiary care hospital-based study has been published. Effect of Moringa oleifera leaves on the hematological profile of fluorosis-affected rats has also been published. Significance of copper, zinc, selenium, and fluoride in squamous cell carcinoma of the cervix, a pilot study has been published. Chronic fluorosis, a disease of concern, is also been published. So these are some of the articles that has got published. And in this mysterious mineral fluoride, we have observed that simple methods for prevention of fluorosis, in addition to the impact of fluoride on various disease outcomes. In this study, estimation of serum fluoride and renal parameters in diabetic nephropathy, a facility-based observational case control study. The major observations that we have made is evaluation of serum fluoride and renal parameters will help in early diagnosis and management of diabetes and diabetic nephropathy, understanding modalities for better patient care, and we have proposed the certain regulations to the Government of India, Ministry of Health and Family Welfare, as well as to the Government of Karnataka, yet to be finalized. A cross-sectional study of serum and urine fluoride in diabetes and fluoride-exposed population. The outcome of this study is that serum and urine fluoride estimation helps in assessing the progress of the diabetes, landing up with microvascular complications, particularly the nephropathy. We have done one more study which has got published, that is estimation of fluoride and SERTIN. This is a PhD-related work in patients with diabetic nephropathy in Kolar district of Karnataka, India. We have observed that alterations in serum SERTIN1 levels indicate the severity of the damage due to stress during hyperglycemia and fluoride toxicity. So this is another study which has got published in the research report of fluoride. serum fluoride levels as a biomarker in pre-eclamptic women, a case controlled tertiary care hospital-based study. Again, this is a PhD-related work. We have observed that fluoride is an added risk factor for preeclampsia, and it has been observed that exposure to more than 3 milligram per liter can be considered as a has got an impact. In our study, we have observed that the maternal serum fluoride level more than 1.8 milligram per liter can be considered as a diagnostic biomarker for predicting preeclampsia. So, till date, what the myth that has been there, that when the people are exposed or when the population is exposed for more than 3 milligram per liter for a duration of minimum of 5 years, there is an impact. This has been observed that with more than 1.8 milligram per liter exposure itself, people can have adverse effects with respect to preeclampsia. Effect of Moringa oleifera leaves on hematological profile of fluorosis affected rats. We observed that the powder of Moringa oleifera leaves, which are grown in this local population using the same water which the people are consuming, has been observed that this leaves powder is effective in reducing the plasma fluoride content, maybe because of the observations that we have made. Maybe because of the observations that we have made that the Moringa oleifera has got a repellent activity against the fluoride that is there in the water or in the soil. Moringa oleifera helps in improving the hemoglobin percentage and RBC counts. The significance of copper, zinc, selenium, and fluoride in squamous cell carcinoma of cervix, a pilot study. The outcome that we have observed is that plasma copper to zinc ratio and fluoride was increased in squamous cell carcinoma study subjects. Plasma zinc and selenium was decreased, but the p-value was not significant. Statistically, it was not significant. The plasma fluoride levels were significantly elevated in squamous cell carcinoma patients. Chronic fluorosis is a disease of concern. This is a review research article that we have done, and this has summarized the major deleterious health problems caused by the fluoride, and which we are going to publish it in the newspaper as well as in the media shortly. This emphasizes that fluoride mitigation needs to be practiced widely, and fluorosis is a preventable disorder. With a simple thought and commitment, it can be avoided. To conclude, to say a few things, it has been observed and documented that genetic factors play a critical role in pathogenesis of chronic fluorosis. Clearly, the mechanism of chronic fluorosis still need further research. Prevention of chronic fluorosis is important. It can be prevented by keeping fluoride intake within safe limits. Important to consider total exposure, that is exposure through air, food, and water when evaluating adverse health effects of the fluoride. The take-home message that I wish to give here is with our experience, the epigenetic and genetic factors in fluorosis affect the human body from womb to the tomb. So with this brief on the fluorosis updates that is being done with respect to the fluorosis research and referral lab, so I wish to say thank you to one and all for your patience here. Thank you.
Thank you, sir. Thank you, sir, for your wonderful talk and sharing your research experience on this mysterious molecule. Now I request Dr. Venkateshwarlu Ravi, sir, to enrich us with his rich research experience on radiation and DNA damage. Over to you, sir. Slides show.
ఇంకోసారి ఇస్తామని Good afternoon, one and all present here. First of all, I would like to thank the organizers of this national webinar on fluorosis and genetic disease, a missing link. Here I am going to talk about the work which I did during PhD in Sri Ramachandra Institute, Chennai. So the presentation outline of my presentation is, so the DNA damage I'll be talking about, and then gamma H2X marker. It is one of the marker for DNA damage and our findings in our lab, and gene and microarray expression marker and our findings in the gene, lab from the Sri Ramachandra Institute of Higher Education and Research, Porur, Chennai. As we all know, the DNA is a genetic material which is present in the inside the cell and is involved in the transfer of genetic information from one generation to another gen, and also it is involved in the functions of the cells. Okay, so the DNA inside the cell in a day-to-day life undergo the damage due to several DNA damaging agents. All these together called genotoxic agents. Here you can see in the slide there are some mentioned. So the DNA replication stress, oxygen radicals, ionizing radiations, chemotherapeutic agents, UV light, and several molecules which leads to the damage to the DNA. So in general, the damage can be divided into several types, which is base mismatches, single strand breaks, and cross-linking DNA double strand breaks. In a normal cell, the damage which is caused by these agents, it will get repaired by using different types of DNA repair mechanisms. If it is a base modifications, it will be mismatches. The mismatch repair will work. And if it is a single strand breaks and base modifications, base excision repair will be there. If it is any cross links, the nucleotide excision repair will be there. In case of double strand breaks, there are two different repair mechanisms are present inside the cell. Those are homologous recombination and non-homologous end joining repair. So NHEJ, we used to call it as a error-prone repair mechanism. So what will be the fate of the cell after this damage and repair? So there are mainly three possibilities is there after the exposure to these genotoxic agents. So first thing is, as you can see, the repair and then be normal. So in that case, there won't be any problem. So in case of improper repair and then incorporation of abnormalities, cell will be alive. In that case, in that condition, it will be leads to the chromosomal abnormalities as well as further that leads to the cancers and other disorders. So the third possibility is the cell, if it is unable to repair, it will undergo cell death. So in that condition, the tissue will get damaged and it will be pathetic. Cells will be dying. So in case of my PhD studies, what I majorly concentrated on DNA double strand breaks due to the ionizing radiations. So I'm from next slide onwards, I'm talking about only the ionizing radiation and the damage which is caused by the ionizing radiation. So you can see in the slide there are ionizing radiation. The human beings are continuously exposing the ionizing radiation from several sources. Here I have mentioned few. One is background radiation, another one is fluoroscopies, and then next CT scans. These three more majorly contributes the low dose radiations, whereas LINAC linear accelerator, which is used for radiotherapy in case of nuclear reactor accidents and then nuclear bombing, where we can, the surrounding people who are working in the medical facilities or in the vicinity of the nuclear reactors, they will get exposed. So here we need, in case of these medical facilities, the people who are working in the facility, they will be monitored by using the personal monitoring devices. This is TLD badges. Whereas in case of higher exposures, means large scale exposures due to the nuclear accidents, nuclear bombing, there won't be such measurement. The facility is available to measure the dose in case of healthy healthy people. So in that condition, we need to, first thing is, we need to segregate from the exposed to unexposed. And another one is, after in the exposure, who is received less dose, who is received higher dose. These are the two things which is required for the medical management of the radiation exposures. So biodosimetry, I am going to talk about some of the markers after exposure to radiation. And then radiation triage is nothing but segregation. So here you can see in the slide, after exposure to radiation, so radiation is nothing but energy when it is passed on the cells. Cell has several molecules, macromolecules and micromolecules. These molecules will get ionized and changes will happen. So all these changes is called radiation markers. So here you can see the changes will be the cellular level changes, chromosomal level changes, protein level changes, and molecular level expression changes, and some of them are biochemical changes. So a biomarker is nothing but it's a measurable indicator of exposure. But what we want is to measure the dose, the marker which need to be useful for the estimation of the dose. That's what we want in this condition. So from past, I think from 1960s onwards, there are several markers they identified which are specific to the radiation, and they established to measure the dose in case of accidental exposures where there is no physical dosimetry is available. So here are some of the markers which are highly established. They are dicentric assay, as you can see in the first image. In general, the chromosomes will have only single centromere, whereas in case of exposure to radiation, the chromosomes will get damaged and it will appear as a two centromeres in the same chromosome. By counting these number of dicentrics, we can relate it to the dose. It is a dose dependent manner. As the dose increases, the number of chromosomes, the dicentrics will increase. Another marker is micronucleus assay, where we can see this is a binucleate cell. It is at the phase of cell division. So some of the genetic material will be detached from the main nucleus and appear as a small portion that is called micronuclei, with small nuclei. So it's also one kind of marker which is used for the measurement of the dose in case of radiation exposures. Another marker is a fluorescence in situ hybridization assay. Here also you can see, we will use the fluorescent molecules, tagged molecules, to identify the damages which are caused by the ionizing radiation. So these three markers require a culture of the cells to bring the cell into the metaphase, where it will require more than 3 days, 4 days to report. But in case of accidental exposures, we don't have that much time. So still research is going on, and then some of the markers which are used for the estimation of the dose is identified. Is one marker is gamma H2X assay, and another one is gene expression assay and microRNA expression assay. These are emerging markers which has a high potential to measure the dose in case of accidental exposures. So during my PhD, I worked mostly in the gamma H2X assay. So as you can see in the image, it's a gamma H2X. It's a histone molecule 2, which is a core molecule in the nucleosome. You can see this H2A will get phosphorylated at the site of the double strand break. Whenever the double strand break happens, whether it might be the radiation or whether it might be any chemical, any agent, any genotoxic agent which cause the double strand breaks, this gamma H2X, the surrounding H2AX molecules will get phosphorylated. So this phosphorylated H2AX molecules, several molecules will get phosphorylated in the vicinity of the double strand break. So we will use the antibody which is having tagged with fluorescent molecule, and we can visualize in the nucleus. This is a cellular nucleus where you can see as a spot. We call it as a foci. It's called as a gamma H2X foci. By counting this number of gamma H2X foci, we can relate this number with the dose. It is a dose dependent manner. So exactly the molecule which get phosphorylated is serine 130 in the H2AX molecule. So I think it's this gamma H2X is first identified in the NIH, NCI NIH Cancer William Bonner Laboratory. After that, it has it's used in several applications. As you can see, it can be performed in several types of samples. Those are lymphocytes, splenocytes, buccal cells, bone marrow, skin, plucked hairs, and some of the cancer tissues also. So because of this, it's a rapid method when compared to the conventional things. So it is being used to measure the double strand breaks in case of different applications. One is radiation trace, radiation biodosimetry in basic research in case of DNA repair mechanisms and DNA replication stress, and also radio sensitivity of the radiotherapy patients, and monitoring some of the drug treatments, epidemiological studies to monitor the toxicities, genotoxicities, and then some of the genotoxic evaluation of the chemicals. So all these studies highlights certain things, missing points. Those are called, what is the baseline frequency without exposure to any kind of radiation? What is the frequency of in the basal level? And also what are the variables influencing the baseline frequency, and also inter-individual variations and methodological variations. From all these studies, these are certain things which need to be addressed. So for that purpose, I worked as a JRF in a DRDO LSRB projects, where we worked majorly on standardization of the gamma H2X in a blood samples, expose, and also find the variables like age, sex, and lifestyle factors, which is influencing the frequency at the basal levels. In the second step, what we did is we collected a healthy volunteers blood sample and exposed in vitro, and then we in vitro to X-rays and gamma rays, and I counted the number of gamma H2X foci. And in the final stage, we want to check whether in case of accidental scenarios in real, so we choose cancer patients who are undergoing the radiotherapy. So we collected the blood samples from them, and also we measured the change, the number of gamma H2X foci in those blood samples. So I'm presenting the results what we got it from these first is gamma H2X baseline studies. So here what we collected 130 healthy volunteers blood sample, age ranges between 18 to 77, and we performed the gamma H2X assay by using the immunocytochemistry. And also the baseline frequency of from all these samples what we obtained is 0.042 plus or minus 0.001. So further we did a subgroup analysis to know whether any significance differences are there. So first thing is we analyzed, is there any difference between the male and female? So as you can see, we didn't find any significant difference in the gamma H2X foci frequency in the basal levels in between male and female. Further, we want to check whether the alcoholic and smoking, is there any influence on the double strand breaks, and measured by using the gamma H2X assay. So here also we find there is no significant difference, but some other reports where they studied it has an alcoholic and smoking has a significant influence on the gamma H2X foci. So another subgroup analysis what we did is we divided all the 130 healthy volunteers into students, laborers, and administrators, and then agriculture works to find is there any significant variation is there in these people. So we found that there is a significant variation among these groups of the gamma H2X foci in the basal levels. So further, we want to see the influence of age. Is there any age influence on the gamma H2X assay? As you can see in the graph, so the gamma, as the age increases, the gamma H2X foci levels are increased gradually with a R-square value of 546. So further, we want to confirm what the results what we got is whether it is similar to the findings from the other laboratories in the contemporary labs throughout the world. So we found that the frequency, as you can see, this is the baseline frequency from different studies. So here what we found is 0.042, and the range is 0.005 to 0.158. The baseline frequencies what we got is in the range similar to the published reports. So these findings we published in a journal, Radiation and Environmental Biophysics, in the year 2016. So further, so we know that gamma is to X basal levels, and also we want to apply this for the radiation measurement of the dose in case of accidental exposures. For any marker, we need a dose response curves to measure the dose. For that purpose, we collected the blood samples from the healthy volunteers, 5 individuals, and then we exposed the blood samples to the different doses ranging between 0 to 2 gray by using the 6 MV Linac X-rays, which is used for the radiotherapy, and also the irradiations procedure as per the IAEA 2011 manual. So, and then we measured the gamma H2X after 30 minutes of the exposure. So here are the the dose response curve, dose response relationship of the gamma H2X we observed. So as you can see, in case of without exposure, there is no gamma H2X foci inside. This is a nucleus where there is no foci, where in case of as the dose increases from A to E, the number of foci is increased gradually. It shows that the gamma H2X response after X-rays is a dose dependent manner, so that it can be used for the application of the biodosimetry. So here are the dose response curve, which is obtained by scoring the 100 nuclei from the each dose. So x-axis shows dose, y-axis shows gamma H2X foci. As you can see, as the dose increases, the gamma H2X foci increases, and dose dependent increase of the gamma H2X after exposure to 6 MV Linac X-rays, which can be used. This dose response curve can be used. The dose response curve coefficients are important, where in case of unknown exposures, we can input Y. So the yield from the unknown samples, and we can get the dose levels we can get. So further, we want to see, is there any technical difference, technical variations are there by using the different microscopes. We used 2 different microscopes. These are epifluorescence microscopy and confocal microscopy. As you can see this graph, in all these doses, the gamma H2X foci obtained in confocal microscopy is little higher compared to the epifluorescence microscopy, because the confocal microscopy we can get images in a sections, different sections, okay, compared to the epifluorescence, but the difference is not significant. So we conclude that we can use both instruments with slight differences, which can be utilized for the dose estimation. So these results, the dose response curve and dose relationships details after X-rays and gamma rays, and also we measured by using the not only microscopy, by using the flow cytometry also we measured, and then these results we published in a cytometry part A journal. So further, we want to check this gamma H2X assay whether it can be used for the real-time exposures. That is, we choose a radiotherapy patients as one of the model, where we collected the blood samples after first fraction. That is, generally they will be giving 1.8 to 2 gray as a partial body radiotherapy. So we collected around 30 the blood samples from 30 cancer patients of different head and neck cancer, breast cancer, cervix, and abdomen. So we collected the blood sample 3 ml pre and 30 minutes post fractionation, and then performed the gamma H2X assay, and then estimated the dose by using the dose response curves, which we already established. So what we found is, so in all these patients who are undergoing the radiotherapy, that is 1.8 to 2 gray of first fraction, as you can see in the figure. So in all these cases, the gamma H2X foci frequency is increased after first fraction. It suggests that the gamma H2X assay can measure the damage which is caused to the lymphocytes is a normal tissue. So here we can relate it that it can be suitable to measure the exposures in case of radiotherapy. So further, we want to relate this increase in the gamma S2X foci frequency with the physical dose, which is calculated based on the some of the CT parameters, radiotherapy machine parameters. There, we collected some of the information from the CT, radiotherapy machine. There's a CTV, PTV. By using the formula, we measured the EWBD, that is equivalent whole body dose. Okay, so in case of this equivalent whole body dose is higher in case of cervix cancer patients compared to the HNC breast and abdomen. So further, we correlated the physical dose and then the dose which is obtained by using the gamma H2X assay. As you can see in the image, so the estimated dose, that is by using the gamma H2X assay, and the physical dose which is obtained from the instrument, that is EWBD, equivalent whole body dose. So we can see the correlation as the EWBD increased, the estimated dose also is increased. It shows that the dose which is delivered, we are able to measure by using the gamma H2X marker. So further, we compared that with in vitro and pre-irradiation and post-irradiation. You can see this, most of the cases, it is higher in case of post-irradiation. So the estimated dose using the gamma H2X assay was correlated with the physical dose, that is EWBD. So with this, the findings of this particular gamma H2X assay, that is baseline studies and the factors influencing the baseline studies, baseline frequency of gamma H2X assay, in vitro dose response dose, and then the suitability in case of real exposures in the cancer patients. We generated the information which can be used in case of accidental exposures, and is there in case of radiation accidents. So further, even though this gamma H2X assay is a advanced and fast compared to the cytogenetic methods which I mentioned, dicentrics, micronucleus, and fluorescence in situ hybridization, there are certain limitations there. Lack of technical expertise in scoring, and also the fluorescence is short-lived. After a few days you can't measure, and also the sensitivity is less compared to the whatever the existed already established markers. So further, we want to look for the another markers. So they are gene expression assay and microRNA expression assay. So in recent years, the gene expression and microRNA studies are increased in all fields of research. So we want to see whether we can use this expression of these gene and microRNAs for the estimation of the dose and in case of biological dosimetry. So for that purpose, we collected the blood samples and then we exposed in vitro, and then we measured the gene and microRNA expression. Here are some of the genes which we look into is, these are ferredoxin reductase, FDXR, CDKN1A, cyclin dependent kinase 1 inhibitor 1A, DDB2, DNA damage specific binding protein, and MDM2, mouse double minute, growth arrest DNA damage inducible alpha protein gene, GADD45A. These are some of the genes which are involved in the DNA repair. Once after the cell is exposed to radiation, the DNA will get damaged. To repair, these genes are involved in the repair process of DNA damage. So we choose these things because already these are reported in some of the studies in the biodosimetry laboratories throughout the world. So we take up these few markers, few genes for the whether these can be utilized for the estimation of the dose in case of unknown exposures. So for that purpose, we didn't look into the baseline levels. We first want to see whether the markers showed a dose dependent increase or not. So for that purpose, we choose another marker is miRNA 101. It is also expressed after radiation. It is one of the DNA damage specific marker. So along with these three genes, we also measured that miRNA 101. So the doses chosen is for the in vitro dose response to 0.5, 0.5 gray, 2 gray, and 4 gray. So we also measured, apart from the in vitro dose response, we measured the gene and mRNA in the cancer patients undergoing the radiotherapy. So you can see in that slide where you have the dose-dependent increase in the fold changes in the particular gene, FDXR, CDKN1A, GADD45. Similarly, the microRNA 101 also showed a dose-dependent increase. So these are initial results. Also, we got the dose response curves, as you can see here. For each one, we have a dose response curve. So further, we want to after constructing the dose response curve, we want to see in the cancer patients. So we collected in the same blood samples which we collected for the gamma H2X, we analyzed the fold change difference in these FDXR, CDKN1A, GADD45, miRNA 101. So what we found is, so 24% of the, in most of the samples, so 76% of the samples, the FDXR gene is high, fold change increase was observed. Similarly, CDKN1A 88%, GADD45 76%, miRNA 101 is 80% of the samples. We have 88% of the samples. We are able to find the fold change difference in these expression of these gene and microRNA expressions. Further, we correlated the gene expression with the EWBD, that is equivalent whole body weight. So 24% of the samples showed GADD45, 24% FDXR, 42 in case of CDKN1A. But it is not exactly correlated with the EWBD. So we predict certain things we missed while performing. So those might be some of the factors which are responsible for the these lack of correlation with the physical dose. We collected the blood samples from healthy volunteers and then irradiated, constructed the dose response. So I think it might be good if we collect the blood samples from cancer patients and then generate the dose response curve. This is what our speculations. Okay, so instead of, we used a relative gene expression instead of absolute gene expression. Also, apart from these things, there will be a different individual sensitivity to the radiation and also the time points. So the gene expression will vary from time to time after exposure to the radiation and DNA damage. So the time point what we choose is 4 hours. So, but might be 16 hour, 24 hour, 48 hours also some of the studies reported. So we might have tried in the other time points also. So, and also whole blood samples instead of isolated and the variability in lymphocytes. These are the some of the things which might contribute the lack of this EWBD correlation with the gene expression and microRNA expression studies. These results we have published in a journal, Radiation and Environmental Biophysics in 2019. So with this, I conclude the presentation and I acknowledge the funding from DRDO, LSRB, MAJRF in that project, and CSIR, SRF for supporting fellowship, and also my colleagues, Dr. Shafa Bashiruddin, Tamit Selvanji, Bhavani M, Karthik Kanagaraj, Sangamitra, and project students who supported me throughout this process. So there, this is the lab where I have worked. So I also thank my RSC members, Dr. P. Venkatachalam, J. Arunakaran, and guide Salman F.D. Paul for helping me throughout this research work. So thank you all. Any questions?
Thank you, sir, for your exemplary talk. Now the session is open for the discussion. You all can ask if you have any questions with sir. Both Dr. Shashidhar sir and Venkateshwar Ravi sir is here with us.
As there are no questions, I request Dr. Soumya to take it further.
Thank you all. Now I request Dr. Sharad sir to give the conclusive remarks. Over to you, sir.
Good afternoon, everyone. I thank the organizers for giving me this opportunity. And we have had two long lectures today, and wonderful and exhaustive. One on the role of fluorosis and few other environmental chemicals in health, and other one is on the role of radiation in health. So what I would like to basically highlight is that the human body that we see is a consequence of the genetics interacting with the environment. So because of this, today we have seen how genetics interacts with the chemical components of the environment and also the physical or the radiation component of the environment. And we have also seen how when these parameters are abnormal, when the radiation is high, or when there are some chemical components in the environment which are toxins, they affect the human health and cause disease. So this is an area which is increasing in importance day by day, because we currently live in a consumerist society where our consumption of material and the environment that we live in has become highly modified. But we are not clear what exact role all this new environment that we live in has. For instance, the toothpaste that we use today is rich in fluorine. Most of our homes have granite flooring, granite tabletop, and granite is known to give away small amounts of radiation, not huge amounts, but we have not understood what exactly is the impact of all this when they are being used day in and day out. This, the environment that we live in is totally different from what our ancestors, just as recently 50 years ago, how our grandfathers lived. So with this, I think we have had a wonderful two-directional inputs, one from the role of fluorosis and other minerals, environmental toxins, and another one from the side of physical radiation. So I think I would like to conclude by saying that we have opened new chapters to understand the role of these components in the new era of human health. I thank the organizers again, and I also congratulate the speakers for their wonderful presentation. Thank you very much.
Thank you.
Thank you, Sharad sir. Now I take this opportunity to propose vote of thanks on behalf of my department and institution. I thank the trustees and administrators of the academy, department colleagues for having attended the webinar. Hope it is fruitful. Thank you one and all.
Dear participants, thank you for your attendance. The feedback form shall be shared with all the registered participants. Once the feedback form is filled, the certificates shall be auto-generated. Thank you one and all.